Hello everyone, if you are new to my channel, this is Fazna Imran and this is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Welcome to Pearson Bio Academy. And currently we are focusing on Pearson Edexcel International Advanced Level Paper Reference WBI 1401 Unit 4, which covers energy, environment, microbiology and immunology. And specifically today we'll be tackling questions from photosynthesis, the papers from 2020 to 2024. According to your past paper question, I have made some further categorization, the topic photosynthesis, and they are structure of chloroplast, absorption and action spectrum, chromatography, photosynthetic reactions, and finally photosynthesis products. And today we'll be focusing on structure of chloroplast. Before moving into the question part, we will see some general idea about the structure of chloroplast. In recent years, you have been asked to do or complete the structure of chloroplast. So let's break down the key components of the chloroplast one by one. First, you have the membrane. You know, the chloroplast has a double membrane structure consisting of an outer membrane and an inner membrane. Together, these membranes are referred to as the surface membrane, and the same configuration you can see in the mitochondria also. Next, we have the intermembrane space. The space between outer and inner membrane is known as the intermembrane space. The stroma is the fluid-filled inner compartment of the chloroplast where various biochemical process takes place. And within the stroma, there is a membranous system called the thylakoid membrane, which is essential for photosynthesis. And a stack of thylakoid is known as granum. Then we have the starch granules. So they serve as storage for starch molecules. And you can see in this picture, chloroplasts have their own ribosome, allowing them to synthesize their own proteins. And the chloroplasts have their own DNA strand, and you can see they typically found in a loop form. Finally, lipid droplets. In recent past papers, they have introduced the presence of lipid droplets in chloroplasts, which are involved in lipid storage. And you know there are membranes in the chloroplast. If you look closely, you can find two types of membranes, the surface membrane and the thylakoid membrane system. In the surface membrane, we include both the outer and inner membranes, while the thylakoid membrane system is separate. And you should be able to recognize the difference between these structures as each has a unique role within the chloroplast. First, we will talk about the surface membrane. If you look at a chloroplast, this outer layer is the surface membrane that surrounds it. This membrane is made of a phospholipid bilayer similar to the cell surface membrane. The surface membrane contains protein channels such as porins, and also they contain glycoproteins which help recognize different molecules. There are also ion channels and other proteins. These channels allow ions from the plant cell cytoplasm to enter the stroma inside the chloroplast. For instance, ions like magnesium and iron are transported because they are essential for synthesizing chlorophyll. Since the structure of the outer membrane and inner membrane is similar, they both appear smooth, but their functions are different. The outer membrane is smooth and permeable to most ions and small molecules. In contrast, the inner membrane also appears smooth but has transport proteins that regulate what enters and exits the chloroplast. The outer membrane allows many more molecules to pass through compared to the inner membrane, but the inner membrane is more selective, allowing only certain molecules into the stroma. Next, we will look at the thylakoid membrane system, the inner part of the chloroplast. You can see here, we are going to explore the structure of the thylakoid membrane. And this membrane looks like a bag structure with a matrix or lumen inside covered by the thylakoid membrane. And inside the lumen there is nothing, just like an empty membranous bag. And this is where the hydrogen ions will accumulate during the photolysis. If we look at the membrane itself, it has a phospholipid bilayer similar to the surface membrane of the chloroplast. The thylakoid membrane also contains photosystem 1 and photosystem 2 where the chlorophyll pigments are attached. Additionally, it has the enzyme ATP synthase which produces ATP, and we will discuss this biochemical reaction in another section. The thylakoid membrane also has electron carrier proteins just like PQ, PC. You don't need to remember these specific names, just keep in mind that they are electron carrier proteins. So if you have already learned photosynthesis, you know we can divide the process into light-dependent reactions and light-independent reactions. In a chloroplast, these reactions happen in different locations. In questions, they might ask you where each reaction takes place. The light-dependent reaction occur in the thylakoid membrane system as these reactions rely on chlorophyll. The chlorophyll pigments are attached to the thylakoid membrane, which is why this is where light-dependent reaction happen. On the other hand, the light-independent reactions takes place in stroma. The enzymes needed for light-independent reactions are found in the stroma of the chloroplast. Then we'll move on to the photosystem 1 and photosystem 2. Let's summarize what's in your student book about this. It mentions that the photosynthetic pigments in the photosystem absorb light in two different chlorophyll pigments, and with each system containing slightly different combinations of chlorophyll. And this difference is very important because it means each photosystem absorbs light at slightly different parts of the spectrum. PS1 absorbs at a wavelength of 700 nanometer, while PS2 absorbs at 680 nanometer. The book also mentions their locations. PS1 is located in the intergranular lamellae and PS2 is in the granular regions. And these details are also very important. Let's move to the question first, and the first question from the 2020 January paper. In this question, you are asked to explain the role of chloroplast pigments in the light-dependent reactions of photosynthesis. This question actually falls under the biochemical reactions of photosynthesis, where we'll learn about the chlorophyll pigment. I'm introducing this question here just for your information. So as you will see when studying the light-dependent reactions, chlorophyll absorbs light so that the electrons will be excited within the photosystems, and this excitation leads to the release of high energy electrons which are then transferred through a series of carriers in the electron transport chain. As a result, reduced NADP and ATP, the main products of light-dependent reactions, are synthesized. Don't worry if you are not familiar with light-dependent reactions yet. I will go over this question again under the biochemical reactions of photosynthesis. The next question is from 2020 October paper. This question is straightforward. You have to find out the row of the table show where the light dependent reactions and light independent reaction takes place. It's quite simple. Answer C. Light dependent in thylakoid membrane and light independent in stroma. The next question is from the October 2020 paper. In this question, you are provided with a diagram showing the outline of a chloroplast, and you are asked to draw and label any three features on this diagram. But however, be careful to avoid draw or label the stroma and the thylakoid membrane, as these are not allowed. And if you label either of these, and you will not receive any marks, so make sure to read the instructions carefully. And for the marks, you can draw intermembrane space, starch granules, DNA strand, and finally ribosomes. Be sure to draw each feature clearly and label it accurately. The next question is from the June 2021 paper. You are asked to compare and contrast the structure of chloroplast with the structure of a mitochondria. Let's look at the pictures first. As you may remember, this is the structure of a chloroplast and this is the structure of a mitochondrion. You can see they share some similarities along with some differences between them. So both chloroplasts and mitochondria are double membrane organelles, and they have their own DNA and ribosomes to synthesize proteins. Now let's look at the differences. Remember, we are focusing on structure, not function. If you look carefully, you will see that chloroplast is specifically adapted for photosynthesis, while the mitochondria is adapted for cellular respiration. In the chloroplast, you will notice the stroma and the thylakoid membranes, while in the mitochondrion, there are cristae formed by folds in the inner membrane and also the matrix. Additionally, chloroplasts contain chlorophyll pigments within the thylakoid membrane, which mitochondria do not have. And chloroplasts also have starch granules for storage, while mitochondria lack these granules. So, to summarize the answer, both are membranous organelles which have double membranes surrounding them, and also both contain its own DNA and ribosomes. However, chloroplasts have some specific structures adapted to perform photosynthesis, such as thylakoid membranes and stroma, while mitochondria have structures such as cristae and matrix adapted to perform cellular respiration. Additionally, chloroplasts contain starch molecules and mitochondria do not. The next question is from the October 2021 paper. In this question, you are provided with a diagram of chloroplast. The statement reads, the structure of a chloroplast is related to its role in photosynthesis. And the structure in the diagram are labeled alphabetically from P to V. The first question asks which row in the table identifies the structures labeled P, Q and V in the chloroplast diagram. And based on what we have learned about chloroplast structure, you know P is a ribosome and Q is starch grains and V is DNA. You can refer to the table to match these labels with the correct description, and your final answer is D. This is the second part of the same question, which structure contains a GALP, and GALP stands for glyceraldehyde 3-phosphate, a compound produced during the light independent reactions of photosynthesis. As you know, the light independent reactions occur in the stroma of the chloroplast, where enzymes needed for these reactions such as ribosco are located. So GALP, the main product of the light independent reaction, is produced in the stroma, and in the picture stroma is indicated by the letter R. Therefore the answer is B, the stroma. This question relates to your IAS Unit 2, where we cover magnification. Here we are given with the actual length of the chloroplast as 7.5 micrometers. To calculate the magnification of this diagram, you will need the image length of the chloroplast as well. The formula we will use is magnification is equals to image size divided by actual size. To find the image size, the length of the chloroplast should be measured in the diagram. So first you have to draw a line across the chloroplast, and here it measures 10.7 centimeters. But remember, the units need to match. So we will convert this measurement into micrometers. Since 10.7 centimeter is equals to 10.7 into 10 to the power 4 micrometers, we can substitute it into our formula. Dividing 10.70 into 10 to the power 4 micrometer by the actual size of 7.5 micrometers gives a magnification of about 14,267 times. And for more questions like this related to Unit 2, I have uploaded a separate video. I will include the link below if you had like to check it out. The next question involves structure T and U, both of which are described as membranes. We need to compare and contrast these two structures. Please make sure to give your answer in paragraph format rather than using bullet points. Let's first identify what each structure represents. In the image, structure T refers to the thylakoid membrane, while the structure U represents the inner membrane, also known as the surface membrane of the chloroplast. Now let's revisit what we have learned about these two membranes. Looking at the thylakoid membrane T, we observe that it has a flattened disc-like shape in contrast to the smooth outer shape of the inner membrane. And both membranes share some similarities. You can see they are composed of phospholipid bilayer and they contain some types of proteins. For example, thylakoid membrane contain ATP synthase and electron carrier proteins, while the inner membranes contain some protein channels. However, their differences are also notable. The thylakoid membrane contains chlorophyll and other photosynthetic pigments necessary for photosynthesis, and they also contain electron carrier proteins and ATP synthase. On the other hand, you can see the inner membrane lack of these photosynthetic components because its main function is to facilitate ion transport into the stroma of the chloroplast. And if we summarize these points, you can give your answer like this. Both U and T are made up of phospholipid layer, but T forms flattened disc-like structures while U is smooth. T contains photosynthetic pigments and ATP synthase, while U does not. Additionally, electron carrier proteins are attached on the T but not on the U. And additionally, region marking scheme emphasizes that both membranes contain proteins. In this question, you are provided with an investigation that explores how periods of light and darkness affect chloroplast DNA levels in one species of a single-cell organism. And you have to read the introduction very carefully to understand its setup. In this experiment, the organism was divided into two groups. One group of these organisms, Group A, was exposed to 36 hours of continuous light, and the other group of these organisms, Group B, was exposed to 12 hours of light, followed by 12 hours of darkness, followed by 12 hours of light. The graph shows the results of this investigation. Here you can see the graph. It's essential to examine the question before analyzing the graph. The question notes that this organism undergoes cell division through mitosis, but cell division occurs only during dark periods. And this detail is critical because it implies that cell division is timed with periods of darkness separate from chloroplast DNA replication. And finally, the question asks you to draw two conclusions about the replication of chloroplast DNA based on the graph. By first reading the question carefully, you can approach the graph with a clear direction, focusing on how chloroplast DNA replication is influenced by light exposure and the timing of cell division. Let's begin by analyzing the graph. The x-axis represents time in hours, while the y-axis shows the mean level of chloroplast DNA per cell. Starting with Group B, in the first 12 hours when exposed to light, you can see the chloroplast DNA levels increase steadily. After 12 hours, the group is placed in darkness, and we can observe that chloroplast DNA replication stops during this dark period. However, mitosis occurs in the dark. So it means the chloroplast DNA replication happens independently from the cellular division. And when we keep the group B after a light exposure, then you can see again the DNA of chloroplast is started increasing. And in contrast, group A was exposed to the continuous light for the full 36 hours. Here, chloroplast DNA replication occurred without any interruptions, leading to a consistent increase in DNA levels throughout the period. This analysis shows two key conclusions. Chloroplast DNA replication only occurs during periods of light as seen in both groups. And also chloroplast DNA replication is independent of cell division since DNA replication halts in the dark despite ongoing cell division. So you can give your answer like this. The replication of chloroplast DNA does not occur in the dark. Therefore, light is needed to the replication process, and their replication is independent from the cell division of the organism. The next question from the January 2022 paper asks you to explain the advantage of having different types of chlorophyll pigments in plants. To start this, let's recall the absorption spectrum, which illustrates how different pigments absorb light at various wavelengths. For example, you can see chlorophyll A primarily absorbs lights in blue region and the red-orange of the spectrum. And chlorophyll B absorbs light in the blue and red regions, but at slightly different wavelengths than chlorophyll A. And if you look about the carotenoids, they absorb mainly in the blue-green region and contribute by covering other parts of the spectrum. So each pigment absorbs light at different and specific wavelengths, meaning that altogether they can cover a broad range of the visible light spectrum. So this wide range allows plants to maximize light absorption with minimal light waste. And if you summarize these points, you can give your answer like this. The presence of various pigments allow the plant to absorb a wider range of light wavelength. Each pigment absorbs light most efficiently at different wavelength. Therefore, more light energy is absorbed, so the rate of photosynthesis will be maximized. The next question, also from the January 2022 paper, provides a table with information about various structures in chloroplast and asks us to identify them based on their functions and with the given additional information. Structure P, this structure is associated with protein synthesis and we know it's ribosomes. Structure Q, this structure is described as colorless and is the site of light independent reactions and you know it's stroma. And structure R, this is the compartment where hydrogen ions accumulate and this function points to the thylakoid membrane. Recall that thylakoids are bag-like structures and the space inside called the lumen is where hydrogen ions gather. Finally, S, storage granules. So these are likely to be starch granules which store energy for the plant. The first question asks where in the chloroplast Rubisco is active. So referring to the diagram, Rubisco is active in the stroma, which is where the light independent reactions occur. Therefore, the correct answer is structure Q. The next question asks what is stored in structure S, referring to the diagram. Structure S represents storage granules, specifically starch granules, so which store starch molecules in the chloroplast. So your answer is C, starch. The next question asks where GP, glycerate 3-phosphate, is formed. According to the diagram, GP is produced in the site of light-independent reactions which occurs in the stroma of the chloroplast. Therefore, the correct answer is structure Q. The next question asks for the correct order of size from smallest to largest for structures P, R, and S based on the diagram. To accurately compare the sizes, we must first convert all three structures into the same unit. If we do not convert them, we cannot determine the correct order. For structure P, given as 20 nanometers, to convert these into micrometers, we multiply it by 10 to the power minus 3 and it's 0.02 micrometers. And for structure R, given as 4.35 into 10 to the power minus 4 millimeters. To convert this to micrometers, we multiply it by 10 to the power 3 and it's 0.435 micrometers. And for structure S, it given as 35 micrometers, which does not require any conversion. Now comparing the sizes in micrometers, P is 0.02, R 0.435 and S 35. Therefore, the correct order from smallest to largest is PRS and the correct answer is A. Remember, converting to the same unit is essential for making accurate comparisons. Again, you have to complete the structure of chloroplast. You can draw any three labeled structures except outer membrane and stack of thylakoids because they have already provided you. You can draw inner membrane and intermembrane space, starch granules, DNA strand, intergranule lamellae. The membrane connects two stacks of thylakoids, ribosomes, and finally lipid molecules. The next question, same as the we discussed in 2021 October paper. Here also you have to compare and contrast the structure of the outer membrane of a chloroplast with that of a thylakoid membrane. And in the previous question, we do a comparison in between inner membrane and the thylakoid membrane. You know both outer and inner membrane share similar characteristic. Therefore, the answer is the same here also. So you can give the answer like this. Both outer membrane and thylakoid membranes are made up of phospholipid layer and contain proteins. Thylakoid membrane contains photosynthetic pigments and ATP synthase, while outer membrane does not. Additionally, electron carrier proteins are attached on the thylakoid membrane but not on the outer membrane of chloroplast. The next question is from the January 2023 paper, and it's a particularly interesting one. This is not about chloroplast structure but focuses on photosynthetic bacteria. The statement explains that while some bacteria are photosynthetic, they do not have chloroplast. The diagram shows a photosynthetic bacterium, and the first question asks, which label structure contains the gene coding for enzymes involved in photosynthesis? Looking at the diagram, you can see that the answer is R, as R represents the DNA loop. Next, the question asks where the light-dependent reactions occur. In chloroplast, these reactions take place on a membrane with attached pigments. Similarly, in this bacterium, it would likely occur on T, which is the cell membrane, as this may contain pigments necessary for these reactions. Although these details might not be covered in theory, with your knowledge, you should be able to tackle questions like this. Lastly, we are asked where the light-independent reactions take place. In chloroplast, these reactions happen in the stroma where the necessary enzymes are present. Here, in the absence of a stroma, the light-independent reactions would take place in S, which is the cytoplasm of the bacterium. So your answer B, S. The next question also very easy. It's related to the structure of chloroplast, and here also the structures are being labeled the alphabetic order from P to V. The first question is, which is the site of photolysis? So photolysis will happen on the thylakoid membrane. Therefore, your answer is T, and the correct answer here is C. The next question, which contains a gene coding for rubisco. You know, in chloroplast, they have their own DNA set which codes for the proteins necessary in photosynthesis. So the answer is V, the DNA strand. And the next question, where do hydrogen ions accumulate for use in photosynthesis? You know, the ions accumulate in the lumen of thylakoid membrane. So its answer S, here it is C. The next question is quite easy. How many of these structures labeled P, T, U, and W are made of phospholipids? If you look at the diagram, P is a ribosome, T is the thylakoid membrane, and U is the inner membrane, and W is the outer membrane. Therefore, 3 structures out of them were made of phospholipids. So answer C here. And the final question, it's actually related to the IAAS topic, but it's very simple, which describes a phospholipid. If you can remember the structure of phospholipids, they have 1 glycerol molecule and 2 fatty acids, and they are differ from other lipid molecules. They contain 1 phosphate group instead of 1 fatty acids. So the answer is A. Glycerol attached to 2 fatty acids and 1 phosphate group. Here is a quick summary of the past chloroplast related questions by year, which may help with spotting patterns on predicting what could come up in future exams. 2020 June asked about the role of chloroplast pigments. In 2020 October focused on locations of the light dependent and light independent reactions and included a question requiring you to draw the chloroplast structure. In 2021 June, comparison between chloroplast and mitochondria. In 2021 October, emphasized chloroplast structure and particularly comparing the thylakoid membrane with the inner membrane, and included a question analyzing a chloroplast, a DNA replication graph. 2022 January asked about the advantage of having different types of chlorophylls, and 2022 October required a drawing of the chloroplast structure and the differences between thylakoid membrane and the outer membrane. In 2023 January, questions focused on photosynthetic bacteria structures, and 2023 October revisited questions about chloroplast structures. For 2024, neither the January nor May papers covered chloroplast-related questions, which means there may be a chance these topics could appear again in the next exams. Likely predictions include questions on drawing chlorophyll types or perhaps more about the structure of the chloroplast, since they have not been asked recently. I hope this video was clear and informative. Thank you for watching Pearson Bio Academy. If you haven't subscribed yet, please hit the subscribe button and don't forget to give this video a like. We'll be tackling more questions on photosynthesis and other key topics in the future. So stay tuned. Thanks again and see you next time. Bye.